Hello, everyone. Good morning or good afternoon, depending on where you are at in the world. Welcome to today's webinar titled Advancing Immunotherapy Testing with Functional Antigen-specific T Cells. My name is Sydney Panilla, and I am with LabRoots. I will be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and brought to you by RoukenBio. To learn more, visit rouken.bio. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, you may also use the Ask a Question box to let us know, and we can assist with troubleshooting there. Okay, I would now like to welcome our speakers for today's webinar, Kevin King, Senior Scientific Advisor, CGT at BioIVT, and Emilio Cosimo, PhD, Scientific Director of Innovation, RoukenBio. Emilio, I will go ahead and hand it over to you first. You may now begin your presentation.
Welcome everyone, and thank you for joining today's webinar, Advanced Immunotherapy Testing Functional Antigen-specific T Cells. I am Emilio Cosimo, and I serve as the Scientific Director at RoukenBio. My background is in oncology and translational research, and I'm deeply passionate about bridging the gap between cutting-edge science and clinical application. Today, we'll explore the rapidly evolving landscape of immunotherapy, focusing specifically on both the scientific advances and the practical challenges of harnessing antigen-specific T cells for drug discovery and therapy. I'm delighted to be joined by Kevin King. We are a contract research organization based in Scotland, in UK. We are located approximately 20 miles from Glasgow and Motherwell. We have more than 110 employees and constantly growing. In terms of expertise, we are a world leader in human immunology research service provision for the assessment of immuno-oncology, autoimmunity, and biosimilar therapeutics. We apply a unique blend of immunology, bioanalytical, and molecular biology expertise to establish advanced experimental solutions to assess the safety and efficacy of novel therapeutics. We specialize in antibody-based drugs and advanced medicine, including immunotherapies. We support a global customer base who operate from the earliest phase of drug discovery to the clinic. We offer a vast array of standardized and also bespoke in vitro models for T, B, and NK cells, myeloid cells, and Tregs, enabling mechanistic studies of immune cell function, therapeutic efficacy, and safety. Engineered cell banks and primary cell assays support reproducibility and translational relevance. Our immune cell therapy platform encompasses construct design, viral, non-viral, CRISPR, transposons. We have an array of immune cell engineering expertise from alpha, beta, gamma, delta, NK, and macrophages. We have scalable production, G-Rex Baractus, for example, and a comprehensive characterization for cytometry, cytotoxicity, and potency assays. And finally, our vision is to be the most trusted research partner for biotherapeutic innovators. At RoukenBio, we combine deep expertise in immunology bias development with a commitment to rigorous reproducible data. We focus on solving complex challenges and accelerating IND-enabling studies across inflammation, autoimmunity, and immuno-oncology. By delivering quality data and fostering collaboration, we help innovators move confidently from discovery to clinic. So here's our roadmap for today. In part one, I will provide a scientific overview of the antigen-specific T cells, discuss the main challenges in immunotherapy, and present a case study on T cell exhaustion. I also introduce RoukenBio's capabilities and how we're working to overcome bottlenecks in this field. In part two, Kevin will discuss the importance of high-quality starting material and donor management, critical factors for reliable cell-based assays. As we go through the agenda, I invite you to consider which topics are most relevant to your work and where you see the greatest opportunities for innovation. Then we'll wrap up with key takeaways and a Q&A session. Immunotherapy has transformed cancer treatment, but significant hurdles remain. One major challenge is T-cell exhaustion, which is driven by chronic antigen exposure and limits the effectiveness of therapies like CAR-T and checkpoint inhibitors. Moreover, tumor microenvironment is another key barrier for therapeutics, as it's a complex immunosuppressive ecosystem that includes regulatory T cells and suppressive cytokines, making it difficult for T cells to function and infiltrate. Tumor heterogeneity and also mutations and antigen loss can lead to relapse even after initial success. And finally, cytotoxicity presents safety concerns and can limit applicability of these therapies. So altogether, these main challenges, I believe, characterize current immunotherapies. And in this context, I believe the antigen-specific T cells are essential for testing new drugs and useful for modeling exhaustion. Autoimmunity presents a different set of challenges. Here, autoreactive T cells become activated due to a breakdown in immunotolerance, leading to the immune system to attack the body's own tissues. Therapeutic strategies may target T cells directly, as in T cell-driven diseases such as MS and type 1 diabetes, or B cells in conditions involving autoantibodies, like SLE and rheumatoid arthritis. In fact, innovative approaches such as CAR-T therapy are being explored to eliminate pathogenic cells, including the repurposing of CD19 BCMA-based CAR-T therapies for autoimmune diseases like, as I mentioned, lupus and rheumatoid arthritis. Antigen-specific T cells, I believe, are crucial tools for developing therapies that restore tolerance without compromising immune defense. Antigen-specific T cells, or ASTCs, are a cornerstone of the adaptive immune system. Each T cell expresses a unique T cell receptor, or TCR, enabling to recognize specific antigen presented by the MHC or HLA complex. These cells are vital for fighting infections, eliminating cancer cells, and in general maintaining immune memory. Understanding antigen-specific T cells biology is key to developing targeted therapies for cancer, infection disease, and autoimmune disease. Advances in T cell engineering are opening new avenues for treatment. However, the low frequency of these cells in peripheral blood remains a challenge for both research and clinical applications. I would love to hear from you how you currently track or utilize these type of cells in your work and what challenges you faced in sourcing and expanding these cells. Okay, let's break down the main T cell subsets. So we have the CD4 positive helper T cells that coordinate immune responses by activating B cells, macrophages, and through the cytokine secretion, recognizing peptides presented by HLA class II molecules. Then we have the CD8 or cytotoxic T cells directly killing infected or malignant cells, recognizing peptides presented by the HLA class I. And also a fundamental category is the regulatory T cells, or Tregs, which maintain immunohomeostasis and self-tolerance by suppressing excessive immune response. And lastly, but not least, memory T cells, usually CD4 positive, persist after infection to enable rapid responses upon re-exposure. Understanding this subset is crucial for immunotherapy, as manipulating their balance can enhance efficacy or safety. For example, boosting cytotoxic T cells may improve cancer clearance, while expanding Tregs could help treat autoimmunity. The diversity of T cell receptors is vast, up to 10 to the 15 possible combinations, but only about 10 to the 6, 10 to the 7 are present in any individual. Adults have around 4 times 10 to the 10 CD8 positive T cells, but each antigen-specific clone is relatively rare. This low frequency makes it quite challenging to start utilizing these cells in research and therapy. And tracking these rare cells is crucial for understanding disease and improving diagnostics and treatments, therefore. So having scalable sources of antigen-specific T cells is critical for drug testing, drug discovery, and also modeling exhaustion. And this is why standardized cell banks are so valuable. Now, let's focus on TCR-T cells, which are T cell engineered to express specific TCR for targeted therapy and research. This approach allows us to create cells with defined specificity, overcoming the rarity of natural occurring antigen-specific T cells. TCR-T cells are a powerful tool for both therapy and drug discovery, enabling us to test immunomodulators and model disease. T-cell therapies involve engineering a patient T-cell lymphocyte usually to express antigen-specific TCR, enabling recognition of intracellular tumor-derived peptides presented by the MHC molecules. Unlike CAR T cells, which target surface antigens, TCR T cells can access a broader range of tumor-associated antigens, including cancer testis antigens and neoantigens here depicted. Here I wanted to show you this specific paper, which highlights the recent progress in actual solid tumor indications, not just hematological ones, focusing on pancreatic cancer with the KRAS mutation. However, there are challenges for these therapeutics. There is the specificity issue, which is essential to avoid off-target toxicity. Persistence is needed for durable responses, and continuous antigen exposure can lead to T-cell exhaustion, as mentioned. Additional TCR therapy is constrained by HLA restriction, which relies on tumor MHC expression in patient HLA type. Right. CAR-T and TCR-T are both adoptive cell therapies, but they differ fundamentally. CAR-T cells recognize surface antigens independently of MHC, allowing a broader patient applicability in rapid, potent killing, whereas a TCR-T recognizes peptides presented in MHC, enabling targeting an intracellular protein by requiring HLA matching. This diagram summarizes the differences between CAR-T and TCR-T, which already mentioned previously. But one thing to mention here is also the fact that CAR-T is a much more high clinical maturity compared to TCR-T, having 7 approved therapeutics compared to only one recent one, 2024, by Adaptimmune. And we believe that the TCR-T are not only a valuable therapeutic modality, but also a valuable tool for testing immunomodulating therapies designed to either boost or suppress immunotherapy. And as mentioned before, a major challenge is having sufficient numbers of antigen-specific T cells for real-time drug evaluation. To address this, we created transgenic TOE-and-GO TCR-T cell banks that provide a scalable, ready-to-use solution for high-throughput screening. We believe this platform can accelerate drug discovery by enabling rapid and reproducible testing of immunomodulators. At RoukenBio, we developed a robust and scalable method for generating TCR-T cells. The process starts with donor selection based on HLA genotype, supported by our partners at BioIVT, ensuring that the engineering TCRs will recognize their target antigens in the right immunological context. We then use retroviral vectors for stable TCR expression. And our expansion protocol is optimized for both yield and functional quality. Each batch is well characterized, not just for transaction efficiency, but also for markers of activation such as CD137, interferon gamma, and exhaustion like LAG3, PD1, and TIM3, to include some, which are critical for predicting in vivo performance. This approach allows us to create large and consistent banks of TCR-T cells, enabling both research and high-throughput drug screening. We then assess the functionality of our transgenic cells. In this case, I'm showing you the MART1 specific TCR-T, and we had two systems. We measured the cytotoxicity using the melanoma-derived MIWO cells, which express levels of MART1, and you can see they're highly potent compared to non-transduced T cells. And also we use a T2 cell system loaded with a MART1 peptide or control peptide, and where we can see the activation confirmed by detecting CD137 at the surface or production of interferon gamma, demonstrating an antigen-specific response. These assays validate the potency and specificity of our TCR-Ts, and the functional validation is critical for the risk in translational projects and supporting perhaps regulatory submissions. And we ensure that cells perform as intended in relevant biological contexts. We use TCR-T cells in the context of immunomodulators in a high throughput 384 well assay system. We use target T2 cells, pulse with antigen of interest, and expose them to TCR-T cells in presence of increasing concentrations of T cell inhibitors, which are the test articles. And in this case, we can show here, we have both test articles reduce activation markers in the surface as CD137 and supernatant as detected by interferon gamma. This demonstrates the utility of this platform for autoimmune discovery, drug discovery. It is essential to have large cell numbers per vial to enable robust screening of multiple compounds. This approach allows for rapid identification of novel therapeutic targets and or biomarkers, supporting both basic research and drug development pipelines. Now, let's focus on endogenous antigen-specific T cells, which essentially are T cells with the naturally occurring TCRs, expanded without genetic modification. This approach preserves biological relevance and can provide important insights into natural immune response. Endogenous T cells are especially valuable for studying immune response in infectious disease and autoimmunity, where the natural repertoire is critical. At RoukenBio, we can actually expand naive CD8 positive T cells with endogenous specificity using a proprietary protocol with artificial APCs, which is distinct and different from the more conventional monocyte-derived APCs type of expansion methods. We also conducted functional activity by culturing with peptide pulse HLA matched target cells. And we believe this is a useful tool and preserves biological relevance and avoids introducing secondary TCRs. As an example here, I want to show you some data regarding CMV restricted T cells from donor PBNC supported by our collaborator BioIVT with high levels of purity and yield. Activation is measured by, as before, by CD137 and interferon gamma production after exposure to the CMV peptide pulse target cells. This approach is applicable to other antigens and disease models, making it valuable for studying vaccine response, immunomemory, and the impact of immunomodulators in a physiological relevant context. And here is just a schematic of all the essentially key differences between an endogenous antigen-specific T cell and a transgenic TCR-T and a CAR-T here. Now, I want to shift the focus on T cell exhaustion. Understanding overcoming exhaustion is critical for improving the efficacy of T cell-based therapies in cancer and also in chronic infections. Here, I will present our capabilities of RoukenBio in this context, and then I will also present a case study on modeling antigen-specific T cell exhaustion. First of all, let's introduce what T cell exhaustion briefly. T cell exhaustion is a complex multifactorial process that has profound implication in immunotherapy. It's characterized by progressive loss of effector functions, upregulation of inhibitory receptors such as PD1, CTLA4, TIM3, LAG3, and distinct transcriptional changes such as TOX is the main driver, TCF1 reduction, and NR4 increase. Importantly, as anything T cell biology, exhaustion is not binary. It exists as a spectrum, and some exhausted cells can be recovered, reinvigorated with checkpoint inhibitors, while others cannot. And understanding the molecular underpinnings of exhaustion is key to designing therapeutics that can either prevent or reverse it, depending on the context, if it's cancer, for example, or autoimmunity. And let's move on to our current methodology, RoukenBio. RoukenBio, we established an in vitro model for generating exhausted T cells using repeated antigen stimulation, depicted here. This assay can be combined with secondary functional assays, such as mixed lymphocyte reactions, or MLRs, or cytotoxicity assays. It's applicable for testing immuno-oncology therapies, including checkpoint inhibitors and bispecifics. Having a standardized exhaustion model is invaluable for drug screening, allowing for reproducible assessment of candidate therapies across multiple donors and experiments. Let's delve a little bit deeper on the data sets of our model. Our T cells in exhaustion model, we have upregulation of multiple checkpoint proteins such as PD-1, LAG3, TIM3, and CTL4, but also costimulatory molecules such as CD122 and CD137. Not shown here, we also have the tox. Factor tox is also upregulated. And here you can see the overlay between dark blue, which is unstimulated, the slightly lighter blue, which is stimulated once, and then the multiple stimulations and exhausted, essentially, phenotype of the T cells for our model. And upregulation of all these markers is generally considered as a hallmark of T cell exhaustion. As mentioned above, at RoukenBio, we generally have two main approaches for modeling exhaustion, or essentially testing therapeutics that might interfere with the T cell exhaustion. We have the, after repeated multiple stimulations, we have more of a classical T cell dependent cytotoxicity, or TDCC, on the left here, for example, against the cancer target, and then measured by impedance. On the other side is more of a classic mixed lymphocyte reaction, MLR, where you have two donors, one for the monocytes derived dendritic cells, and the donor B for the T cells and exhausted model. And then you will co-culture the two, adding the therapeutic of interest, and you can see changes in the phenotype or cytokine release. And those two models can really be used in tandem, or depending on your research question and specific context of your indications and modalities. So let's focus more about the MLRs. In this case, I'm showing you a classic MLR using our exhausted T cells, which were co-cultured with an allogeneic immature monocyte-derived dendritic cells. And then we after the incubation, we measured several things, such as the proliferation capacity by Ki67, which was dramatically reduced, and then some cytokines compared to, everything was compared to conventional and non-exhausted T cells in dark blue. As you can see, all these factors were reduced dramatically, including interferon gamma, GM-CSF, IL-2, and TNF-alpha. And this functional impairment is reproducible across donors, donor pairs, and experiments. We believe that this assay can be used to screen for agents that restore T cell function, providing a robust platform for drug discovery. And again, within a 5-day MLR settings type of experiment, blocking PD-1 with nivolumab partially restored here, as you can see, interferon gamma production in exhausted T cells, increasing concentration of the drug compared to the isotype control. However, you can determine that comparing the exhausted T cells versus non-exhausted conventional T cells, the order of magnitude of increase is dramatically reduced. This suggesting it is only a partial rescue. This, I guess, indirectly might suggest the limitations of checkpoint blockade and the need for combination strategy, perhaps. So understanding this limitation is crucial for designing effective immunotherapies and accelerating drug discovery in this case. Now focusing on the PD-1 axis blockade, using either PD-1 blocker, nivolumab, or PD-L1, atezolizumab. Either of those increase proliferation of exhausted T cells in, again, MLR reaction. And this is, again, measuring percentage of Ki67 positive, CD8 positive T cells. So you can see compared to isotype control, Both types of strategies have a positive impact on proliferation capacity. And this clearly demonstrates the potential of checkpoint inhibitors to restore T cell function, but also underscore the need of optimizing dosing and scheduling for maximal benefit. But the main whole message of this is that this model is a really attractive system to screen a therapeutic of interest. Again, finally, regarding PD-1, we had a look at the PD-1 checkpoint blockade using pembrolizumab, increasing concentration of, and we had a look at cytotox capacity of exhausted T cells and how efficacious in rescuing the exhausting phenotype of T cells, co-culturing them with the SCOF3 in the presence or absence of pembrolizumab, and then measuring cytotox via our impedance system using the exogenous RTCA. You can see that the PD-1 checkpoint blockade resulted in dose-dependent enhancement of cytotoxicity. And this is another example how you can use this methodology, this use model, to screen for the therapeutics and potential novel therapeutics. I would like to focus now on the case study I was talking about, which investigates how chronic antigen stimulation in solid tumor drives antigen-specific T cell exhaustion, which limits the efficacy of T cell bispecific antibodies. The authors cover both mouse and human antigen-specific T cell models to dissect the functional consequences of exhaustion. I will be focusing on the human side for this webinar. And from their paper, similar to our platform, their model uses a MART1 specific CD8 T cells subjected to persistent antigen stimulation, which induces exhaustion, generates these TX T cells, which are compared to cells that have been stimulated once, which are the TF in this case, for the response to the human TRP1 TCP, which is a human T cell bispecific antibody targeting cross-linking essentially T cells with CD3, and TRP1, which is an antigen found in melanoma cells. This is a great approach because it provides a translational relevance by modeling the clinical scenario of chronic antigen exposure in cancer. And as I said, I will be focusing on the human side of the paper, which is essentially one figure. So I'll focus on this. The study demonstrated that MART1 exhausted T cells show markedly reduced cytolytic activity against TRP1 positive cells after TCB engagement compared to the actual acutely activated T cells. And you can see the difference between the red line, which are the exhausted T cells versus the non-exhausted, which are either with or without the TCR engineered, are much more responsive in green and in blue. Then the figure moves on to detecting the granzyme B expression or cells, T-exhausted cells that are positive for granzyme B. As you can see, there is a reduction for both donors, highlighted in red compared to the blue and green lines. So there is also a lower response in terms of granzyme B expression. And finally, they also measured diminished levels of secretion of key pro-inflammatory cytokines, here as interferon gamma and TNF-alpha. You can see between the difference between the green and blue line and the red, and cytotoxic proteins such as perforin and granulosin upon TCB stimulation. These results were consistent across two healthy donors, underscoring the robustness of their assay and model. This highlights the importance of functional validation of novel therapies in preclinical models, which can inform clinical decision-making and therapy selection. I would consider the potential for integrating exhaustion models and biomarkers into your own research pipeline to accelerate discovery and improve translational outcomes. So, to summarize this case study, this paper, or the specifics of this paper, I think they show that chronically stimulated antigen-specific human T cells provide a robust and reproducible in vitro model for exhaustion. Because the phenotype emerges from antigen persistence, it captures the biological context in cancer and/or chronic infection. When we pair this model with complementary readouts, real-time cytotoxicity, cytokine profiling, flow cytometry, and transcriptional programs, we get a multi-angle view of T-cell fitness that is sensitive to both efficacy signals and potentially toxicities. The translational value lies in how these datasets guide decisions. If a drug restores interferon gamma but not proliferation, that suggests a specific mechanism and a rationale for combination therapy. If cytotoxicity improves only at high concentration, that might inform dosing strategy or biomarker-driven patient selection. So in short, this model let us test immunotherapy under realistic conditions before they can be selected or reach the clinic. And that also helps prioritizing the pipeline of a specific drug discovery program. And I want to conclude this part by just summarizing our in vitro exhausted T cell model by saying that these cells are not allergic. They retain capacity to respond, but their response is lower in terms of cytokine production, proliferation, and cytotoxic potential. It is quite interesting to see that blocking the PD-1 axis can partially restore their function, but not completely. And this highlights the fact that there are limitations in monotherapeutics, and perhaps we can use this model for rational combinations, for example, with co-stimulatory agonists, bispecific, or kinase modulators. In practice, this model can be designed for medium to high throughput mechanism of action type of studies. It can be scaled across donors and can accommodate either kinetic or endpoint assays. And we have quite a long experience with this type of models because we were the first CRO to operationalize a human in vitro T-cell exhaustion model that closely enough mimics the tumor microenvironment behavior, which is obviously important to inform drug discovery programs. So what does this mean for your projects and for researchers? We believe that RoukenBio, we can accelerate immunotherapy testing with the ready-to-use, well-characterized thaw-and-go cell banks, which are designed to remove common bottlenecks in sourcing, for example, expansion and standardization. Our offering enable high-throughput, real-time assessment of immunomodulators, reduce variability and complexity compared to custom cell generation, and also support translational relevance across oncology, autoimmunity, and checkpoint inhibitor studies in general. We provide the TCR-T cells and the endogenous antigen-specific T cells both as a service but also as a product, depending on your needs. And our scalable production ensures consistent quality and supply for an R&D development need. And with that, I would like to conclude with a little summary of this part. The first part, I talked about antigen-specific T cells and how essential they are as both as a therapeutic approach, but also as a toolbox for drug discovery, and how they are challenging because obviously they're relatively low frequency. We also address this by resolving this issue by producing large quantities of TCR-T cells and also endogenous antigen-specific T cells. Our exhaustion model is a well-established and valuable tool for screening immunomodulatory agents. And also we experienced that the consistency in donor sourcing, HLA typing, a process is what turns a promising assay into a really reliable decision-making platform for drug discovery. And that's why our partnership with BioIVT is crucial, because it ensures quality and continuity from the very beginning of the development. And that will link with the next step, the next part, which I would like to hand over to Kevin King, the Senior Scientific Advisor in Cell and Gene Therapy at BioIVT. He will share further insights on sourcing high-quality starting materials and donor management strategy, which are fundamental to successful cell and gene therapy programs. Thank you very much.
Thank you, Emilio. It has been a pleasure collaborating with you, and I'm very encouraged by the innovative research and advances that the team at RoukenBio have accomplished. It is exciting to observe the impact your work will have on research and therapies now and into the future. Just a little about me. My name is Kevin King, and I am the Senior Scientific Advisor for Cell and Gene Therapies at BioIVT. I bring more than 30 years of experience in blood banking and cell therapy collections, including founding of Physicians Plasma Alliance and PPA Research Group, which pioneered disease state apheresis collections and immune cell processing. Throughout my career, I have opened and established multiple commercial donor centers and supported BioIVT's expansion from research into clinical collections, both in the US and the UK. Let's begin with some of the challenges we face in providing cellular starting materials. Starting at the upper left, by examining how we establish and maintain a reliable network of highly qualified donors. Many donor registries report having tens of thousands of volunteers. However, the composition of these registries warrant closer scrutiny. Most individuals in such registries initially enrolled as potential bone marrow donors, a generous and altruistic commitment, but they may have never undergone a full leukapheresis procedure, which is the base of cellular starting materials and the most commonly requested product. A leukopak collection typically requires more than 4 hours of time to complete. Prior to the leukapheresis procedure, the donor must pass all eligibility screening, including infectious disease testing, a complete blood count, have at least 2 robust, easily accessible venous access points, and be in good health. In contrast to the other donor registries, to be considered a recallable donor within our database, a donor must have completed at least one successful leukopak donation. This prior donor experience increases the likelihood of a successful collection. Although variability and unforeseen challenges can still arise, this highlights only a fraction of the complexities associated with donor management. Next, as we move across to the right, we turn to the donor matching process. For our qualified donors, we collect a comprehensive set of attributes, including core demographics, HLA typing, Fc gamma receptor profiles, and relevant medical history. In addition, we offer customizable donor characterization panels, which may include expanded viral testing, functional immune cell attributes such as memory potential, metabolic fitness, and other effector function profiles. Back to the bottom left, our optimized collection protocols enable targeted isolation of specific cell populations, maximizing yield and allowing us to provide high-quality starting materials that can model clinically relevant scenarios, which are important for preclinical and translational research. Finally, as we move across to the right, it's critical to engage with a vendor whose operational framework supports seamless progression from research use only material to GMP compliant processes. Vendors can vary widely in capability, and partnering with an organization that has decades of specialized collection experience is essential for reliably supporting cell therapy development programs. We employ a strategic end-to-end approach to support your research and cell therapy workflows. Our process begins with the use of deeply characterized, reliable, and recallable donors with collection capabilities established in the United States, the United Kingdom, and additional global regions. We provide optimized collections that yield fit-for-purpose cellular products, enabling seamless support for our partners operating across international markets. Donor materials are available in both fresh and cryopreserved formats with fully customizable door-to-door white glove logistics services designed to maintain strict temperature control and ensure a timely delivery of high quality products. Among donor-related variables, Recallability represents the most critical operational and scientific attribute. Regardless of the depth of donor characterization or the extent of laboratory testing performed, the inability to reliably schedule a donor at the required time negates the value of all prior analysis. Therefore, the donor recallability is the foundation to ensuring continuity, reproducibility, and the reliability of downstream research and therapeutic workflows. To support this requirement, we intentionally cultivate long-term relationships with our donors to ensure timely availability when products are needed. Recallable donors are defined not only by their willingness to participate, but by their demonstrated reliability and consistency across multiple prior collections. Several factors contribute to donor recallability. First, we maintain a regular structured communication with donors to inform them of the available programs and to reinforce the importance of their contributions to both our operations and our clients' research and clinical objectives. Second, recallable donors have consistently demonstrated punctuality and adherence to scheduled collection times. This is particularly critical given that all leukopak collections typically exceed 4 hours in duration. Delays in donor arrival can necessitate significant operational adjustments, and in the case of same-day shipments, may jeopardize courier schedules and downstream experimental timelines. In addition, recallable donors must successfully meet all standard screening and eligibility criteria at every collection event. Equally important is the donor education. Donors are informed of the scientific and therapeutic significance of their material, fostering a clear understanding of their role as active partners in the research process. This emphasis on education and engagement enhances compliance, reliability, and provides overall collection success. Cell therapy research and developers should regard cellular starting materials as one of the most critical determinants of product quality, consistency, and clinical performance. Correspondingly, raw material suppliers bear the responsibility of designing, implementing, and maintaining robust, well-controlled, and reproducible processes centered on this foundational input. The quality of starting materials directly influences manufacturing reliability, process scalability, and downstream therapeutic outcomes. Navigating the regulatory landscape governing cellular starting materials can be complex, as multiple regulations and guidance documents have been established to support compliance, quality assurance, and industry best practices. These frameworks are intended to guide organizations in building systems that ensure control, traceability, and risk mitigation throughout the product lifecycle. Integrating these regulatory expectations into a coherent development and manufacturing strategy can be particularly challenging for organizations. As part of any process design or development effort, it is essential to reference and incorporate at a minimum applicable sections of the Code of Federal Regulations, such as Parts 1271, 210, 211, and 820, and the EU and UK ATMP regulations, appropriate to the type of product and stage of development. Ultimately, the objective is to establish highly robust, well-controlled, and reproducible processes that consistently deliver high-quality starting materials. These principles align directly with good manufacturing practice expectations and should remain central to both development and commercialization strategies. We collaborate closely with RoukenBio to develop customized collection and processing protocols tailored to their specific research and clinical objectives. These protocols may be designed to enhance targeting of specific cell populations, or when required to intentionally model other collection conditions that reflect challenges observed in a clinical setting. Such flexibility enables more accurate evaluation of process robustness and therapeutic performance under real-world conditions. We are utilizing an advanced software program that analyzes collection parameters and outcomes to support process consistency and repeatability. This system enables data-driven optimization of apheresis procedures, ensuring that targeted collection outcomes can be reliably achieved across multiple donors and collection events. In addition, we have a robust inventory of frozen leukopaks, PBMCs, and immune cell subsets from both healthy and diseased cohorts. We also offer customized cell isolation workflows to align with customer-specific requirements. When standard off-the-shelf solutions do not meet the needs of a particular program, alternative strategies can be developed in collaboration with our scientific and technical teams. Beyond donor demographics, HLA typing, and genotyping, we provide expanded characterization options, including cytomegalovirus, CMV status determination, and other additional IgM or IgG-based serological assays. For non-standard or highly specialized cellular products, we work directly with customers to design and deliver solutions that meet defined technical and regulatory specifications. We understand to be a great partner in the clinic, we also must be a great partner in research. At BioIVT, we offer integrated and scalable pathways designed to support preclinical research requirements for both fresh and cryopreserved starting materials. This infrastructure enables continuity of supply and process alignment as programs advance from early research into clinical development. Importantly, these capabilities extend seamlessly into the clinical phase with equivalent enhanced offerings available for clinical collections, including fresh and cryopreserved GMP compliant leukopaks. This continuity ensures consistency in starting material, attributes, processing approaches, and quality controls across preclinical and clinical stages, thereby reducing translational risk and supporting efficient clinical progression. We support more than 30,000 specimen collections annually, providing extensive operational experience and scalability. Our donor network includes over 600 recallable HLA-typed leukopak donors, enabling reliable access to well-characterized cellular starting materials. In addition, we maintain a unique global network of clinical partners, facilitating coordinated collections across the diverse geographic regions. To ensure material integrity and regulatory compliance, we offer comprehensive chain of custody solutions, incorporating real-time monitoring of both temperature and location throughout storage and transport. Our centralized point of contact model streamlines donor matching, service coordination, and logistical execution, allowing our customers to focus on their scientific and clinical objectives while we manage operational complexities. Our organization is supported by a multidisciplinary team of quality, operational, and regulatory professionals with more than 250 years of combined industry experience. This depth of expertise underpins the design, implementation, and maintenance of a robust quality system aligned with global regulatory expectations. We maintain a strong track record of successful inspections and audits conducted by regulatory authorities, We operate ISO-compliant facilities and College of American Pathologists accredited locations. In addition, our operations are fully aligned with applicable data protection and privacy regulations, including General Data Protection Regulations, GDPRs, and California Consumer Privacy Act, the CCPA. Our portfolio includes a comprehensive range of cellular starting materials available in both fresh and cryopreserved formats. Offerings include normal and diseased leukopaks, mobilized leukopaks, as well as peripheral blood mononuclear cells, PBMCs, and defined immune cell subsets such as natural killer cells, CD8, CD4 cells, CD34 hematopoietic progenitor cells, and these cellular products are sourced from multiple biological relevant origins, including peripheral blood, bone marrow, umbilical cord blood, and mobilized donors. In addition, we offer tissue-derived immune cell populations isolated from secondary lymphoid tissues, including spleen, lymph nodes, and tonsils. This breadth of material enables support for a wide range of immunology, cell therapy, and translational research applications. Practical and operational perspective, BioIVT provides a comprehensive support to address the diverse sourcing and data requirements associated with research programs. Our extensive global network is designed to support studies across discovery, translational, and clinical research stages, and into therapy. We maintain partnerships with more than 400 institutional review board approved collection sites worldwide. These sites encompass a broad spectrum of clinical environments, including local physician practices, donor centers, and large integrated health systems. Through collaborative relationships with our collection partners, we work closely to ensure that human biological specimens are sourced in accordance with defined protocols and accompanied by the specific clinical and demographic data required to support rigorous scientific investigation. We are committed to delivering high-quality biospecimens and the associated data in a timely, reliable manner to support global research and development efforts. Our operations are conducted under more than 40 IRB-approved protocols, enabling a broad range of collection types and ensuring compliance with ethical, regulatory, and regional research requirements. We maintain an extensive inventory of over 800,000 biospecimens and support both prospective collections and remnant specimen acquisitions. This infrastructure enables flexibility in the study design, ensures availability of materials across a wide range of research applications. Our biospecimen offerings include, but are not limited to, normal control specimens, matched sample sets derived from individual donors, support for longitudinal study designs, specimens collected in support of vaccination studies, specialized expertise in blood processing and immune cell isolations, customized design collection kits tailored to specific protocols and analytical requirements. In addition to physical specimens, we provide comprehensive high-quality supporting data packages. And this may include disease severity metrics, diagnostic or pathological classifications, demographic variables, treatment history, relevant medical background information, follow-up clinical outcome data, and molecular and genetic data, including mutation status, genotyping, and next-generation sequencing, and other advanced genomic analysis as applicable. Together, these capabilities support robust study design, data integrity, and translational relevance across discovery, translational, and clinical research programs. In summary, our mission is to consistently deliver high-quality products in alignment with your program timelines and requirements. We are committed to responsive communication and collaborative problem solving to ensure continuity of supply and operational success. I want to sincerely thank you for your time and attention during this presentation today. And again, it's been my absolute pleasure working with RoukenBio, and we're excited to support their development of antigen-specific T cells and other cellular solutions they offer. Thank you.
All right. Wonderful. Well, thank you both for your very informative presentations. We will now go ahead and move into the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. You can just type your question into the Ask a Question box and click Submit. We will go ahead and answer as many of your questions as we have time for.
Yeah, keep them.
All right, let's go ahead and jump right in here. Okay, Emilio, it looks like the first question we have here is for you. And this one says, what are the timelines for generating a new bank of TCR-T from conception to full characterization?
Thank you for the question. I think we roughly around about 30 days from, I guess, the selection of the target and the feasibility assessment. That will include, obviously, if there is a specific donor, in collaboration with BioIVT, if there is a specific donor characteristics, actually typing, obviously, and all that. So we'll be around about 30 days from conception to full bank with a sufficient amount of phenotypic characterization and functional characterization.
Okay, perfect. Next, it looks like we have a question for Kevin here. This one says, Slide 5 emphasizes that recallability is the most critical donor attribute. What is your success rate for recalling specific donors?
Yeah, that's a great question. I mean, we have a very high success rate, mainly the reason is by the time they become a donor in a leukopak, they've done this many times and they're well vetted. However, life happens, donors have work issues, they get sick. There's always that variable. I think it's just mainly we try to be as consistent with follow-up and have backup plans established well ahead of time, because for those instances when donors just can't show up. But it's always important to keep that in mind, but we have very high success rate for donors to be recallable.
Mm-hmm. Okay, perfect. Emilio, this next one is for you. It says, what release criteria matter most for research use banks?
Okay. I think I mentioned in the slide, so we're looking for at least a 50% for the TCR-T cells. We're looking for at least a 50% expression of the transgenic TCR. Then we're looking for multiple cytokines quantifications levels for gamma, interferon gamma, TNF alpha, also including other ones like IL-2. We are looking at phenotypic markers such as activation, such as CD137 and CD69, but also exhaustion markers, which I guess you can the transgenic also expressed during activation, which PD-1, LAG3, and TIM3. So those are the main coordinates, the main parameters we're looking to essentially, I wouldn't say qualify the bank, but at least characterize the bank so that we can pass it for either internal studies or as a soon to be launched kind of products. And also the other thing is, because we have this as a thaw and go, as I mentioned, we're looking at for stability and also post resurrection, post cryo preservation, and see how viable they are. Usually they are above 90%, but we are establishing minimum threshold for that. I hope
Okay,
that answered the
perfect.
question.
Thank you. The next one we have here, back over to you, Kevin. This one says, how do you handle a situation where a reliable donor becomes ineligible?
Sure. It's really critical to discuss these contingency plans up front with our sponsors and partners to establish continuity plans and communicate early. I would say, it's good to keep open communication with donors and the clients. But we do our best to offer alternative dates when things come up, donors just can't donate, or we have multiple backups in line that are HLA tested, matched with the same demographics. So we try to have all that lined up well ahead of time. So when those days occur where donors don't show up for some reason or another, we have those backup plans already well established, and we know what steps we're going to take to move forward.
Okay, great. The next one that we have here says, it says you had a table showing that TCR-Ts may have higher off-target toxicity than CAR-Ts. But if we compare, for example, a CAR-T that targets an overexpressed gene on a cancer cell with a TCR-T that targets a neoantigen, wouldn't the TCR-T be safer?
Guess it's for me. That's an excellent question. I guess that table probably was oversimplifying and categorizing, but I still think, yes, it depends on the context. So in theory, yes, a neoantigen-specific TCR-T should be relatively safe, but there have been occasions of toxicity, sometimes fatal, of essentially not necessarily of tumor on target, but also off target, i.e. cross-reacting with specific antibody, with specific proteins that were not intended really to interact with. And there have been some cases of cardio toxicity and other toxicity in the eye, I think in skin, because essentially the TCR raised against MART1, for example, wasn't really reacting against MART1. So that's the main, I think, issue I have with TCR-T. But if it's well designed and is well characterized during your IND pre-non-clinical studies, you can probably control those toxicities, but you have to be monitoring during clinical trials. I hope that answered the question.
Okay, perfect. The next one we have here is for Kevin. And Kevin, this one says, could you provide matched samples of PBMCs and tumor cells from pancreatic cancer patients?
Yes, we can. I just caveat that with it would be difficult, and maybe the timeline would be extended just because of the rarity of donors. But yes, we could do that for sure.
Okay, wonderful. The next one we have here says, what is the lowest frequency of antigen-specific CD8 T cells you started with and managed to expand for downstream functional assays?
That's a good question. I don't think I have a number specific, but I think it was under the 1%. We're looking at 0.3, 0.4, but I will probably get back to you with a specific number more correctly. And we were able to, as a multi-step expansion, and yes, we were able to develop a protocol to expand enough cells. I think, yeah, it was specifically for CMV, and we were able to expand enough cells relatively similar to the TCR-T banks in order to do a quite relatively medium throughput screening, or I guess in this field you can call it high throughput, but it's not like it's not the same as using small molecule inhibitors or biologics. This is obviously live cells.
Okay, great. We will go ahead and wrap up today with this final question. And Kevin, this one is for you. It says, are the quality standards for your US and UK sites identical?
That's a great question. We're one of the only commercial sites that have this capability across the US and UK. So we standardize definitely where we can. We have integrated critical component solutions that meet the higher of the standards. So if the FDA standards are higher or the MHRA, HTA standards are higher, we always meet and exceed those for both locations. We standardize on strict donor selection and screening, collection and processing, testing and safety standards, training, reporting, and compliance, and certainly ethical considerations, privacy, GDPR, and then it just requires continuous monitoring and improvement, and just watching for changes and guidances across both the networks. But we do keep those standard.
Okay, great. Well, thank you both again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, RoukenBio, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we may not have had time for and those submitted during the on-demand period will be addressed by the speakers via the contact info you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time. Thanks so much, everybody. Bye-bye.
Bye-bye.